Hi everyone. We are welcoming Megan Wyam today, and she's gonna talk with us about linking vocal production and anatomy to acoustic outputs. Thank you.
Thank you so much. Hey. You okay? Thank you so much for having me here. Thanks so much for being here, and thank you to the crew who's running everything, who's been very kind and generous with me. I've had quite a chaotic trip, so it's been very nice to be taken care of, and I appreciate it very much. Okay, so I'm going to talk about linking vocal production anatomy to acoustic output. I'm just going to get right to it so we can not go over time. So sexual selection and vocal diversity is the overarching theme, and it's anatomical mechanisms of vocal production and functional outputs that I am focusing on. And here's a little outline of the talk, starting with acoustic communication and sexual selection, just a small overview, and then examples of this as a case study in bison. And then a comparative study of vocal signal production mechanisms, looking at deer. This is a cross-species comparison of the diversity of vocal anatomy and vocal signals that come out of this. And then the impact on species discrimination once you have hybridization between these species. And then lastly, looking at vocal chains associated with social rank changes in meerkats. And this is sort of an alternative developmental pathway to increased vocal diversity within a species. So what is sexual selection? We all know it's a mechanism of evolution focused on the ability to obtain mates or fertilizations. Through these two main parts of it, the intrasexual selection, competition among individuals of the same sex for access to the other, and then intersexual selection, one sex chooses among mates from the other sex. So generally, the sex that invests the most time, resources, and energy in producing or caring for offspring is the limiting resource over which the other sex competes or tries to attract. Usually, the choosier sex is the female, largely driven on differences in gamete size. However, there's other factors that we really need to consider that are very important in this, that can influence the degree of mate choice and the direction of competition within the sexes. And this is parental care and ecological factors, life history, demography, things like that. Whoops, that didn't work. If you have shared or male-based parental care, increased female competition, things like that. We have an unbalanced sex ratio or variable female mating quality. All of those can influence the direction of this intra and intersexual selection. We also have selection for secondary sexual characteristics, and these are traits involved in courtship or competition, not necessarily reproduction itself. And these often create sexual dimorphisms, and when you have higher reproductive skew, you have stronger selection for these secondary characteristics. Linking this back to communication, you have individuals that make these competition and mate choices based on available information, and this information is conveyed through signals that evolve through sexual selection, which is acting upon the signal, the signaler, and or the receiver. And it often influences the behavior of the receiver. It can be cooperative, but it can be often manipulative or eavesdropping is involved. Quick overview of what types of signals these two types of sexual selection can drive. First, we have intrasexual selection, which is driving signals of fighting ability or condition, can give information on motivation or status, like dominant status. Signals are often held reliable through costs or constraints, and this can be like your proximity risk or receiver retaliation risk. It could be a physical or a physiological constraint that you can't fake this signal. It could be an energetic cost. And often there's an escalation of signals that happen to avoid dangerous conflict between individuals. Then going from the intersexual selection side, there's various mechanisms that can drive this. And I won't go into all of these, but for example, the Fischerian runaway selection or indicator traits, which are giving information on the quality. This can be direct benefits or indirect benefits through good genes or a handicap principle, or sensory exploitation or sensory bias. For instance, here in the guppies, you have the male that has these orange spots that attracts the female, and it's thought that the female attraction is based on her interest in orange as a food signal, and so the male sort of co-opting that and attracting the female based on a sensory bias she has for something completely different. You can also have signals that are driven by trying to signal genetic compatibility between species. important to understand that these are not mutually exclusive. These can be happening, these selective forces, at the same time, and it often depends on the costs and benefits, which can change in space and time, and based on the factors I talked about before, like life history traits, or demography, or ecology. So I'm going to explore how selection can act upon vocal anatomy and its resultant acoustic signals. And although many of these mechanisms and drivers can influence vocal anatomy, I'm going to primarily focus on these, which are signals of male fighting ability or condition, and indicator traits, where signals are giving information about the quality of an individual. So here's a little diagram or a set of pictures that gives an overview of vocal production in mammals, and with deer anatomy as an example. So we have the larynx and the vocal tract. These are CT scans of deer, and the larynx here in the middle, with the trachea coming down. And then here's the top view of the larynx in the upper right here, looking down on the larynx, and an X-ray. And then we have a side view, a lateral scan of the larynx, and here's the vocal folds right there. So we have the cricoid cartilage, which is sort of the shield or the Adam's apple of the larynx, and that protects the vocal folds. And then under it, you have the cricoid cartilage. Oh, wait, there we go, which is a little ring that also protects the vocal folds from underneath. Then you have the arytenoid cartilages, little triangular cartilages, which pull and tighten and loosen the vocal folds to help manipulate frequency generation. And so there's the vocal folds. And then you can see where it would be on this scan. So linking the anatomy and the sound production in mammals, we talk about the source filter theory, which can be applied to other types of species like birds and frogs as well in slightly different ways. The important thing here is that the anatomy influences the acoustic properties, often in very reliable ways. So you have the sound that's generated as air goes through the vocal folds, and it sets them into an oscillation and in the larynx. And that the size of the vocal folds and the structure of the vocal folds determines that fundamental frequency. And this is what it would look like if the sound came directly out of the animal without going through the rest of the vocal tract. You can see there's equal energy within these harmonic structures. But it does go through from the larynx, right there, through the rest of the vocal tract, and as it does so that the cavities of that vocal tract are shaping the signal or filtering it. Some frequencies are getting amplified and others are getting denuded. And yeah, let's see, here is, in Pratt, just a generated tone, harmonic tone with no filter. This is 100 hertz. And then what I did was I spoke into Pratt, and I said a phrase, and Pratt measured my formants, and then took those formants and applied them as a filter to this harmonic tone from above. And that created these formants that you can see here, where some of the frequencies are amplified and others are not. And then the shape of them changes as the shape of my vocal cavity changes as I speak. And this is what it sounds like.
Humans love meerkats.
So you can see how those things are related. So I'm going to give a little case study now on acoustic communication and sexual selection in bison. This is what I did for my dissertation. And bison are large ungulates that have considerable sexual dimorphisms. Here we have a male that's defending a female behind him. The groups are sexually segregated for most of the year. The males are off on their own. The females are in large groups with the calves. They're roaming across the prairie. No territorial defense. But as spring comes, all of a sudden they all get together, and the males start competing for dominance with each other and defending one female at a time as she's coming into estrus with the hopes of mating with her. And then once they do, they'll move on to the next one and try to mate with her. So it's sort of a succession of males guarding this one female until she's ready to mate. And then, so this mating system is female defense polygyny. It's about 6 to 8 weeks in the middle of the the middle of the summer. And it's called the rut. So it's highly dominated by male competition for females, but there could be a role for females. There's just less known about that. There's anecdotal evidence that females will approach other males if they're getting tended by a male that's low quality, and that'll force an interaction and a fight, and maybe she'll end up with the better male. is also evidence of them, well, anecdotes of them running through the herd, which creates a big bison mating storm where they all start fighting, and then hopefully she ends up with the best male in the end, although that can raise a lot of chances for sneaky matings, which certainly happen when the more dominant males are distracted. So there's two mating strategies for the males. The more dominant bulls defend, as I said, this one female as she's coming into estrus. There's the female with a calf that was born that season. And then the other males, either they're challenge him directly for access to that female, or if they're not quite strong enough to take him on, they will just hang out in this little perimeter around him between like 5 to 10 body lengths, and they'll just follow him around, sometimes bellow back at him, but not often. And when he gets tired from other challengers or just he can't forage as much because he's guarding the female and he weakens in condition, then they'll try to challenge him and take over. And so it's sort of this succession of bulls, as I said. This mating strategy of being a satellite bull here is not as reproductively beneficial, but if you're not a strong male, that might be your only choice. So the dominance ranks are consistently changing through the rut because being a dominant male is very, very exhausting, and your condition fades fast. And copulations, or estrus is a very short period for these females, and they usually mate quickly, and then they usually only mate once per season. So it's very valuable to get this one copulation. And so dominance is very important, and it's established and maintained through a variety of signals in close proximity. And there can be visual signals where they'll walk past each other and size each other up. They'll urinate into the ground and they'll paw up the sand on themselves and then they'll roll around in it. The idea being that the testosterone in their urine is getting coated on their body, so that's that musk that you smell during the rut that pervades everything. And so that's a signal that they're sending to the males and potentially the females that they're a high-quality male. And bellowing is a huge signal that you just hear consistently through the rut. There he is. That's what we call a male sexual signal, the bellow. And all of this is to avoid bison fights because bison do kill each other. It's not infrequent when they can't sort out who is dominant from all these other escalating signals. So I'm going to play what a typical bison rut sounds like if you're in the middle of it. Sound like lions or like thunder, and you can imagine when there were millions of them on the prairie, it was pretty unreal. So we want to, so the assumption is that bellows are really loud, costly male signals, and they're probably indexical signals of quality and condition and motivation that are being used by males and potentially females within the rut to increase their reproductive success. So we asked two questions. Do bellows contain important and reliable information about the caller? And is this information intended to by receivers in a useful, functionally relevant way? So throughout the rut, we did behavioral observations of all the agonistic interactions and who was mating with who and who was challenging who. Weight data was collected at the end of the year. So the refuge that we did this work on, they herd the bison into chutes every year to weigh them. And so that's how we can get weight, which is very handy. We collected genetics for parentage information on reproductive success, and we did targeted recordings of tending males, and we also did playback experiments to test out these factors. So here's just a selection of 5 different bison males. So you can be very low frequency. We recorded many, many bison from the back of that truck. You can see me there with my microphone. They sort of ignore us, so we would just kind of sidle up to them, and sometimes they would walk right by us, and sometimes they just stood next to us and bellowing, and it was pretty easy and safe data collection, but it was great to be in the middle of the herd. So from all of these bellows that we collected, we could narrow it down to just under 1,500 that were clean, because as you heard from that first recording of all of bison together, it can be hard to get a really clean recording with everyone calling at once. So from 23 males, we used Pratt to extract some acoustic parameters, and here you can see they're quite low frequency. It can go down to 30 hertz in some cases, and duration is about average 2 seconds. Obviously, there's variation. but they're very loud. So 107 dBc is the sound of a chainsaw or a lawnmower if you're at 1 meter. So we assessed this first question through two different hypotheses. The first one being that these male physical parameters of mass and age would predict format frequencies, and because there's an anatomical constraint there. And then secondly, that these format frequencies would predict male indices of quality, like dominance, copulations, and offspring. So I'm going to focus on formats here, and we found that, yes, mass indeed does predict format dispersion. Heavier bulls have lower formats. And then when you control for weight, we still found that format dispersion predicted copulations. So bulls with lower formats have more copulations, so a higher chance of higher reproductive success there. And, there we go. So if we control for weight, why would there still be differences in format dispersion? But it could be, well, during development, if they have higher testosterone, their vocal tract might grow longer, and so they have differences during vocal development as well as just their overall size of their body, because it can be influenced by hormonal surges and things like that. So just to review, we found a significant result between mass and format frequencies predicting copulations. And when we looked at the correlations between copulations and these other measures of male indices, they were highly correlated. So you can see that it's likely that these bellows are being used to transmit very important information to the receivers. But are they attending to it? So we tested this with playback experiments to tending pairs where we resynthesized bellows and we upshifted them or downshifted them by 2 standard deviation. It's about 44 hertz. And the upshifted formats, here this is just to highlight where some of them, places to look to see where they might be upshifting and downshifting. The upshifted formats simulate a smaller smaller male, and the downshifted formats simulate a larger male. And this is what it sounds like. So we used nighttime playbacks because we wanted the only signal that they were getting to be this acoustic signal, and that they couldn't really tell if there was not a bison behind this speaker that wasn't giving this call. So we thought that would be the most realistic way to get an honest reaction from them. And here is just, I'll play what one of these looks like. This is the female and the male behind her. Playback just started. The male looks up. He's thinking about it. Sand and urinating and wallowing. So these are definite agonistic reactions where he is coming up to this speaker and treating it like a challenger. And sometimes we have them walk all the way behind the car looking for where that other bison is. So in general, males responded with aggressive behaviors more slowly after they heard these low format calls, which were simulating larger males. So when they heard those, they would pause more and they wouldn't have an immediate reaction. But when they heard ones that were simulating lower quality males, like here, they would have a quicker reaction in approaching the speaker and things like that. But we saw no difference in the females for this. And so what could this mean? Functionally beneficial in males, faster aggression to lower quality rivals, and reduce perhaps unnecessarily conflict with higher quality rivals that they wouldn't be able to beat in a contest. But for females, why no difference? It could be that these behaviors were subtle and we weren't able to pick it up. I showed you a good example of what one of these looks like through night vision, but sometimes it was quite hard, especially when the moon isn't out. It could be that the playback wasn't long enough to get the full female reaction. And it also could be, most likely, the female wasn't in estrus yet, so she had less of a drive to be paying attention to what this other rival male is. So we have belloformants are most likely sexually selected, and they're reliable quality indicator traits. And so this highlights the important link between anatomy and acoustic output. So after discussing how sexual selection can act on vocal anatomy and acoustic output, now I want to shift to this second part, which is examining how these types of selection processes on anatomy can result in great vocal diversity. We did this with a cross-species comparison of vocal anatomy and acoustic output in the deer family. And deer are a very diverse family of species. They're widely distributed geographically, and oscillations in the Ice Age meant that they expanded in a lot of radiations, so there's a lot of different species in a lot of different areas, and there's a great diversity of vocal signals. The ones I'm focusing on here are fallow deer, red deer, and sika deer. And it's hard to tell from this diagram, but the red and the sika are quite closely related, and fallow deer are a little bit more distant. These deer all have very similar mating systems, though. The males will defend a harem of females from access of other males, preventing their access to the herd, and females can join the harem or not. The males try to keep them in the harem, but they aren't always so successful. So there's elements of female choice and male competition going on within this species. So here are the three calls. I'll play these. I won't go into a lot about the parameters, but here is the fallow, the red deer, and the sika males. So that last one was the sika, and they're quite small compared to the red deer, but the fallow deer are also quite small. So this red deer is much bigger than the other two, even though you can see from the mean FO, the fallow deer has by far the lowest fundamental frequency, followed by the red, and then the sika deer can get quite high. Now, when we look at the formants, we see in fallow deer, we have very strongly descending formants. Might be hard to see here, but you can. There's these emphasized black bands that are a big downsweep within the calls. You can see that a little more clearly in the red. Here's a downsweep. So their formants are being changed or altered during the call, whereas in sika, we don't see that. Now here's the females for comparison. So sort of similar, kind of sounds like a balloon, air coming out of a balloon. And here, instead of the fallow deer having the lowest call, the red deer female has the lowest call. And it doesn't appear to have a lot of mobile formats going on in these species, in this sex of these species. So we want to look at what are the underpinnings of this vocal diversity, and how does the anatomical diversity mirror what's going on in the calls. So I won't spend a lot of time here, but looking at just the red deer and the fallow deer, where they've had the most behavioral work done on choice and competition within their breeding seasons. If we look at the fundamental frequency in red deer, it's not related to age or weight. However, minimum fundamental frequency does increase with fitness. So if you are a high-quality male, you have a higher fundamental frequency, which is interesting. Fallow deer, on the other hand, they're lower fundamental frequency in high-conditioned males and lower minimum fundamental frequency in dominant males, so the opposite's going on in them. For the formants, both of them, in both species, the heavier or more dominant males have lower formants, and these signal high fitness for them. On the perception side, what was done before was that within species, the males in red deer respond more strongly to low formants, so they're picking up on that. And females prefer the high fundamental frequency and lower formants. So they're preferring things that are linked with higher fitness. In the fallow deer, they found that males were more cautious around low fundamental frequency and formants, which again indicates a higher quality male. So what we did was a cross-species anatomical investigation where I collected a lot of heads and necks from the culls that go on in the parks in the UK, of the royal parks and the various farms that have deer. You know, they're reproducing, they need to reduce the populations within these herds. And so I was collecting anatomy that would otherwise be disposed of. And we did CT and MRI scans on these heads and necks. We did detailed dissections here by taking layer by layer down to look where all the muscle attachments were and the ligaments and everything. And lots of CT scans of larynges. I also collected testes, which is not on here, to try to match testes size with, correlate that with vocal anatomy. And then we're examining all of these different factors. So just a little brief overview of what we found in larynx size and vocal fold length. Between the species, we see that there's a very small larynx in Sika. So here's the Sika deer larynx, the male and the female. And they're very similar in size between the two sexes within this species. But in fallow deer, you can see that there's a lot of sexual dimorphism, and the male is much larger than the female for the size of their larynx, even when it's corrected for body size differences. And in vocal fold structure, we see that here's some histology of vocal folds down here on the lower right. The fallow deer, the vocal ligament here, which is where the vocal fold is essentially, it's long and floppy within the fallow deer. And then in the Sika deer, in the red deer, it gets more compact and defined, and then even more so in the Sika deer right there. So you see big differences in the histology and the shape of the vocal fold itself. So not only the length, but the composition of it. And looking at this table, here, we see that the larynx height, that's this yellow line here, just a measure of the larynx, how far it's coming out from your throat. And then the vocal fold is right here in this red line. For the fallow, in the males and females, we see it's quite a difference, sexual dimorphic difference, that remains when you normalize for body size. For males and females of red and Sika, for these two metrics, they're very similar in Sika, a little bit different in males, but once you correct for body size, so normalize for that, as I said, the fallow deer are still very different, but both red and Sika become very similar between the two sexes. Oops. So we haven't analyzed a lot of this, but we did some excised larynx experiments to try to link the vocal fold length directly with the acoustic output. That's a fallow deer larynx. And just to show you that you can create very realistic vocalizations even without the rest of the vocal tract, here is a Sika deer call from a deer itself, coming from a live deer. And then what we did here was recorded a Sika deer larynx after you put it on a tube and blew air through it and sort of manipulated the vocal folds from the outside by squeezing the larynx. We could do this to it. So there's a nice connection between what the anatomy is and what sort of output you get. I don't think we have time for the video. So we can show it afterwards. It's pretty cool, very floppy, fallow larynx. Yeah. So if we look at the filter and the vocal and the nasal tracts within these animals, we see really interesting differences in larynx position. So there's a lower resting position of where the larynx sits in the neck in fallow and red deer. So here's an example of this. We have Sika deer. There's green or blue arrows pointing to where the larynx is in comparison to the axis vertebrae. And then if you look at the resting position of a fallow deer, here's the axis vertebrae. It's way down here. Halfway down the neck is where it rests normally. And then when they vocalize, they can extend it way down. They have muscles that pull it down towards the chest. And so here it is right here at sort of maximum extension. And so the mobility of the larynx is different between the sexes. Here's a dissection where we have in green the larynx and then the hyoid bone in blue. And the connection between it here, there's a ligament that you can stretch. And in Sika deer, I can fit my little nail in between that gap between the larynx and the hyoid bones, and you can't pull it down at all. So it's solidly attached via a very tight and non-mobile ligament. It's not creating a longer vocal tract. You can't pull it down. But in fallow deer and in male deer, or red deer, they do. You can stretch it down yourself, and you can see it when they're vocalizing as well. I'll show some videos of that. And yeah, we'll just skip right to that. Okay, so here is a fallow deer vocalizing, and look for his larynx, you'll see it bobbing up and down as he calls. And remember when we saw the spectrogram of the fallow deer and it had those formants which were downshifting? That is why it's downshifting, because it's creating a longer vocal tract as it's pulling its vocal folds down, its larynx down, which creates this lower formants because of the physical relationships. Here you can see it there. Do it again. Red deer. And you can't see it as well in Sika deer, like where the larynx is, because they have quite a tight neck ruff. But it didn't go anywhere. So just doing a sum up, of the source anatomy, the larynx, in the red and the Sika are very similar, just sort of scaled differently, but the fallow is very different with the sexual dimorphism and the anatomy of the larynx. The fallow is very floppy, and the red deer and the Sika are much more stiff and shorter. And then sexual dimorphisms, as I said, bigger in fallow males. The dimorphisms are bigger in fallow and smaller in red deer and Sika. So what we have here are potential adaptations for low-frequency pulsed calls in the fallow deer with their vocal production anatomy in the larynx, and then small, tight vocal folds in the Sika, which allows them to make these like balloon-sounding noises in the very high frequencies. So we look then for the influence of sexual selection, and is FO used more in mate competition and choice in fallow than it is for red deer and Sika, because we see these adaptations for low frequency? That's a question to be examined. Then for the filter anatomy, we have the red and the fallow being similar, where they can pull their larynx down, where the Sika cannot. And again, sexual dimorphisms are bigger in fallow and red deer for how low they can get it. Females in those species can still pull it down, but not nearly as low as the males. Or they don't try to. Maybe they can, and they just... No, actually, we've tried to, and the ligament doesn't go that far. Then, smallest in Sika. So these seem to be adaptations for format exaggeration in red and fallow males, which could be influenced by sexual selection. This is encoding meaningful information about how big you are. Even if you're pulling it down and you're exaggerating your size, it's still an honest relationship between how far you can pull it and how big your vocal tract is. You can't pull it beyond your sternum, and if you're bigger and stronger, perhaps you can pull it longer, which still is an indication of your strength. So I won't go into North American elk or wapiti much here, but this is a really interesting species, which is over here. It's actually closely related to Sika deer, even though they're about twice the size as Sika. And they have vocalizations which seem to combine different elements. They have biphonation, which is really interesting. They have a fundamental frequency here, which is generating these low harmonic structure, which gives a nice dense material or spectral area for the formants to be highlighted. But it's also giving this G0, which is an aerodynamic whistle on top of that, which is not related to the fundamental frequency at all. It sort of tracks independently. And here, this one, there's even triphonation in some individuals. So there's a lot of interesting things happening in the anatomy that we've been looking at. And just very briefly on the sources of where this aerodynamic whistle might be coming from, we did some dissections of wapiti and recordings and found that it could be a vortex-induced whistle where we have airflow that's being restricted through this nasal cavity, because when it vocalizes, it sort of pushes its nose up and seems to constrict all of this. So that could be creating constrictions in the passage that make this whistle. Alternatively, it could be a flute-like whistle where air is forced across a narrow constriction that hits the lip of the soft palate. So that is like right here, sort of like replicating a flute. That's still something that is unknown, but it's really interesting to make some guesses based on the anatomy. So here we have unusual adaptations that potentially allow for more diverse communication abilities. So it could be that there's an environmental factor and they need to make sure that their calls are broadcast over longer areas or different types of habitats, and so there's some selection for this as G0. Or maybe it's driven by females in sexual selection, that they like this high quality, or sorry, this high tone, and so males start creating that to be attractive to females. That's something that would be very fun to explore. And just so you can see what it looks like up close when they vocalize. You can see their nose go up. Okay, so we learned that anatomy is a very important proximate mechanism of these signal parameters. And it's important to understand how this signal adaptations are produced with alternative or alterations to anatomy. And this increases our knowledge of anatomy that can facilitate these cross-species comparative anatomy, looking for signal diversity, variation, and complexity. Now let's jump to hybridization. So we can see this increase in diversity within the Cervidae family, but this raises questions on how strongly these signals might serve as reproductive isolating mechanisms because they're so diverse within this family. So when they come into contact with each other, if they were allopatric species that become sympatric, maybe through introductions of humans escaping through zoos, which happened a lot in the UK and other areas, how would these species differentiate between their own species and others? It seems like because the calls are so different, there would be discrimination going on. But when you have breakdown in these mechanisms, then you can have reduction of reproductive barriers and increase in hybridization. So we wanted to look at how these diverse species-specific mating signals function to keep the species apart, and how does hybridization impact this? I'm saying this because hybridization does occur between red deer and sika deer. And sika deer they're not native to Europe or the UK, they're from Asia, but they were in parks and things like that in the 1800s in Europe, and they have since escaped, and in many instances they start hybridizing with the red deer populations that are native. These species have similar reproductive behavior, very different phenotypes, vocal behavior and signals. But the hybridization is bidirectional, so it can happen in either direction, male red deer mating with sika females or vice versa. But most of it happens between female red deer and male sika deer. We know that from the genetics. And hybrids are also physically and vocally intermediate. They're also back-cross easily back into the population, so integration is quite high once you have an initial hybridization reaction or interaction. So here's red sika and then a hybrid of the two. Okay, so we wanted to look at the role that this plays, as I said, in isolation and hybridization. So we did this with doing playbacks to look at the reactions to conspecific, heterospecific, and hybrid male calls. For the red deer, it was in naturalistic conditions in the royal parks in London with semi-wild red deer populations, and we could look at male competition and female choice. Here's a red deer male with a quite large harem of females. And then we also looked at controlled conditions where we would put the females into estrus and then have a choice experiment where we had two speakers and they could choose where they spent their time. We did this with red deer females and sika deer females here in France. So here's the naturalistic conditions. We have our cheekily hid speaker here in the ferns, and then here's the deer. And the first set of experiments, we looked at conspecific versus heterospecific with some controls, the controls being the female sika alarm call and a red deer calf control call. And the second was heterospecific versus hybrid, and then the control here was some pink noise. And the idea is, how are they responding to these calls? Hypothesis that they can distinguish their own species from the other based on the mating calls, and that the males and females will both approach the conspecific male call or treat them like a rival or a potential mate, much more so than the other stimuli. So, significant differences in latency to approach behaviors is what we saw in both sexes. Excuse me. The male responses, they were quicker to approach the conspecific calls over the hybrid calls and the noise, but they never approached the Sika male or the Sika female calls or the red deer calf. The females, on the other hand, were quicker to approach the male heterospecific calls, so they were the red deers approaching Sika males and the hybrid calls more than they would approach the male conspecific calls. And then they were quicker to approach conspecifics over red deer calves and noise. So they would approach conspecifics more than some of the other calls that we produced. Here's an example of female red deer hearing a Sika male.
Start playback.
That's the playback.
Second playback. He's looking now.
I do, there we go, blend it. It took maybe, I think, 5 minutes, but the whole herd got up and moved directly towards the speaker. It's usually instigated by one or two hinds, and then the rest end up following. There they are, doing quite an approach to the speaker to check out what it is. And it's hidden in the, yeah, right there. And they go right up to it. Excuse me. So that was the naturalistic conditions, but what happens when we do these controlled playbacks in the rut, because we don't know that that female or those females happened to be an estrus at that time. Maybe they were just approaching because they were curious, or it sounded, there's some sort of other reason, we're not sure. So here we put females into estrus, they're ready to mate. If they approach a speaker, there's a good chance, or a caller, there's a good chance that a mating is going to occur. And so it's in her best interest to only approach something that she would be willing to mate with. So here we did 2 speaker choice tests. We had a speaker over here on this end, for instance, playing a conspecific call, and then back and forth at the same time with a heterospecific call. And then the female would get released in the entrance gate, wander around, and we would measure how much time she went into and spent in these proximity zones in front of the speaker. The idea, again, that they can discriminate or should be able to discriminate their own species if they're an estrus and only approach the conspecific. But we found, okay, so here we have the 2 different tests, conspecific versus heterospecific speakers going off at the same time, and then conspecific versus hybrid in 2 separate tests. For the red deer females, here we're measuring instance and zones in front of the speaker types, and then how much time she spent in. Out of all of these, there was only one instance one category where she was spending more time in one of the zones, and that was the conspecific zone. She went into more times than the heterospecific zone. But she didn't show any difference in behavior in response to duration or to the conspecific versus hybrid. So she was totally willing to go into each zone and sometimes go directly up to the speaker behind the little screen and see what was happening. So there was not a lot of species discrimination going on in red females for most of these categories. But in Sika females, there was absolutely none. So they would go into each one, and there was no difference in their response in where they were spending their time. So, yeah, and neither of the species preferred the conspecific over the hybrids here. Here's a female that's approaching a Sika deer call, a red female. Again, it was more often that they would approach their conspecific call when they also heard Sika, but there's a lot of variation. Some did go all the way around. Here she is trying to see where this Sika call is coming from, and she goes right up to the speaker and puts her nose on it. Now, she is ready to mate, so you wouldn't think she would do that unless that was something that was interesting. That call is coming from over here, and now the Sika deer is the speaker that's right next to her. Okay, so sorry, there's a lot of text, but just to review, in male competition, the red deer males are faster to approach conspecific males over hybrids, but show no response that a Sika male is a threat to them at all, or a threat to their harem of females. In female red deer, we see that during the rut, they're quicker to approach non-conspecifics, but when we do put them into estrus, they prefer conspecifics over Sika, but when hybrids enter the mix, there's no difference. And Sika deer, oops, there's no difference. Sorry, that got shifted. So it could be that the male response is driven by intrasexual selection forces acting on the vocal tract length, For females, it could be an interest in vocal folds. If females are willing to approach these higher frequency calls, like the hybrid and certainly the Sika. And it could be that the female lack of response is that they're cueing in on other signals to make their mating decision choices, and that they're not actually exerting some sort of driving force on calls within the Sika deer. So the implications are that male calls are not an isolating barrier, and it could facilitate hybridization in some cases. Okay, I know I don't have a lot of time, but I'm going to switch to something totally different. There's only preliminary results, so it'll be a bit shorter. Here we're looking at where we discuss sexual selection and other factors driving diversity, and then how what happens when you have hybridization and how it affects isolating mechanisms. So now I want to explore sexual selection pressures within social rank-related alternative development pathways. So this is within a species where there's change within an individual's lifetime that is impacting its anatomy and creating increased vocal diversity. You can, selection for competitive ability. In the past, it was typically seen as occurring in males as they compete for females, although, as I said in the beginning, this can often change or be variable depending on a lot of other factors that are involved. So in cooperative or eusocial breeders, where reproduction is limited in females, there's stronger selection in females for traits that increase competitive ability. So females are competing with each other in their own dominance hierarchies. In some species, this has led to selection for something called adaptive secondary growth. And this is seen in a range of vertebrates like clownfish, mole rats, and meerkats, where individuals experience growth increases. So they're already an adult, and then they go through some status change like dominance acquisition or changing to be the breeding female, for instance, in mole rats, and they will grow again. So they have this secondary growth phase, which others don't if they don't reach that status. So we wanted to look at what the mechanisms and adaptive significance of this anatomical and vocal changes were in these cooperatively breeding species. Meerkats are a social mongoose living in groups of 3 to 50. If you heard Nicola's talk this morning, I'm sure she told you all about these cooperatively breeding animals where there's a dominant pair living in a group where the rest of the group does cooperative behaviors to help raise the young. Most of the breeding done by the dominant pair, but it's not always the case. There's high reproductive skew, and they're highly vocal with over 30 different call types. Here's contact calls. These are all done in a foraging context as the meerkats are moving through the landscape, digging up food from the sand and eating it. They move as a group, they forage as a group, and here's what their social calls sound like. These are just all different individuals. There's variation in here is linked to a number of different factors, like the presence of pups, behavioral context it's given within the foraging context in general. You know, if they're moving fast or if they're digging in a deep hole, for instance. Social rank can affect it, environmental factors. There's individual and group signatures within these calls. And playback experiments and other types of work have shown that they're used in social cohesion and coordination, and also social monitoring between individuals. So as I mentioned, females show adaptive secondary growth. After a female gains dominance, she increases in her growth rate and her weight and her body length and her head width, so she gets bigger. We don't really see this in males in the same way. This is a graph of it over here. The female is in gray, and 0 point is when she becomes dominant. And you can see this is weight data here, and then all of a sudden it spikes up in females after she becomes dominant. It does increase in males, but if you look across numerous dominance change instances, it's not significant, this increase. So it's not related to age or time spent foraging, this increase in weight. And it's competitive in that this growth is related to rivals. So if you have a rival that's close in size to you and you become dominant, you're going to grow faster and bigger because you need to be bigger than this individual who's very close to you beforehand. And this larger size is functionally beneficial. You're a better reproducer and you're more competitive against other females. Oops, wrong way. So when and where does this anatomical change occur? And is it generalized within certain, the whole body, or is it selectively directed at anatomy that's going to help them increase their competitive ability or reproduction? And how does this change the vocalizations? What are the mechanisms? Is it functionally significant? So we're looking at the first aim is the proximate function and functional effects of dominance. Here, I'm going to skip this just so I'll get to it later. Here's the study of population in South Africa, 350 individually marked animals, where there's lots of life history behavior and very intensive behavioral observations going on. And we have anatomical data and acoustic recordings. So we collected anatomical data at the time of dominance change to try to track them at dominance change, and then 1 month after, 3 months after, 6 months after, to see how they're changing and where this is occurring. So here's weights, here's an X-ray. We anesthetize the animals when we take some of these measurements. Some of this is also done for health checks and to fit collars, things like that, and do morphometric measures. On individuals that die naturally, we take the vocal anatomy and we also freeze the rest of the body for scanning. And here's a CT scan of a meerkat that I did. Here we are, there's the meerkat right there, and we're recording them uber close. So first we want to see, does vocal anatomy and acoustic parameters change after they become dominant? And is this change functionally beneficial? So we tracked these vocal changes within individuals, again, where I said we collected them again and again, and measured these traits. So here's an X-ray of a live individual, and we can measure the height of the larynx and the vocal tract length within it over these time periods. And then we do recordings as well and measure the acoustic parameter change. And then we also measure variables to do with reproductive success. Okay, that's what I just said. And also what I just said. So this, we don't have the data back yet, but have measured X-ray scans for 236 lateral scans. These are lateral on the side, where we're measuring these different metrics of vocal production anatomy. And then dorsal-ventral scans here, where we're measuring head width, but we're also measuring different parts of the anatomy to see where they're putting their energy and if it's growing in other areas which might be beneficial, not just vocal anatomy. And we extracted a bunch of parameters with Pratt. And so preliminary analysis here, looking at baseline. First, here's baseline, when they become dominant, and then 3 to 4 months after. They're tracked within the same individuals. We can see that minimum fundamental frequency decreases within this period, significant decrease. And then there's a trend for mean Q25, so it's sort of a measure of spectral energy, where it's not a format measure, but it can be sort of approximate for measures of that type, spectral density. So there's no patterns due to age or the rival age, and we need to look at weights more in pregnancy and TB. These are dominant females, so newly dominants. If we do the same thing, but with groups, subordinate females in the group that are not going through this change, so far we have 8 measured, but there's more we can do, and there's no significant change in any acoustic parameters so far. So the idea being that they're getting this larger larynx and a lower minimum phenomenal frequency and a larger vocal tract, which could be a lower energy distribution. Then we added on the baseline to 6 months, so this is the same as the first graph before, baseline to 3 to 4, now it's baseline to 6, and we see an even bigger difference. Mean difference before was 37 hertz between these 2 periods, and now it's 50 hertz. So there's an increase in the difference. Most of the change we think happens in the first 2 to 3 months, based on the weight data. We also see that there's, okay, I mentioned that, and Q50, so there's more energy in the lower frequencies. That's significant now after 6 months. And then the harmonic to noise ratio says that there are noisier calls. So we have dominant females, which are lowering in frequency and getting noisier calls, but we need more of the controls to confirm this. So this is something we haven't started. Well, I have all the CT scans, but we're going to do sort of 3D reconstructions of the vocal anatomy and then do geometric morphometric shape analysis on this. So here's the larynx, and then you can get these wonderful full measurements of the size and shape of the hyoid bone. Here's the thyroid cartilage and then the cricoid cartilage. And then look to see how the shape of the anatomy is changing. This is only in deceased individuals, but it'll still give us a good clue if we look at subordinates and dominants, how different their larynx is. And these are collaborators who are doing measurements in other parts of the body, so they measured change in the humerus bone here, and they found positive relationship with bone length and the females who gave birth had higher cross-sectional areas. So here pregnancy is an issue, or it is changing the bones, but rank was not. So it might be that they're not directing this increased size into their humerus bone. That's what I just said. And then lastly, the only slide on this, we're going to do playback experiments to look to see if this is functionally beneficial and are females paying attention to this. If a subordinate female hears a dominant female with a lower frequency call, is she going to pay more attention to that? And here's what the playback stimuli would sound like. So it's the original and then a more dominant call. So it's downshifted in the hertz and then upshifted, be a less dominant individual. So we're studying the mechanisms and functional significance of morphological and acoustic change following this really interesting adaptive competitive growth. It gives us insights into the evolution of signal diversity and vocal development through this new pathway. In some ways, it can be analogous to, say, puberty in humans, where they go through a secondary growth phase in the larynx, where your larynx drops and it increases in size in males more than females. So this creates a wider span of the acoustic signal within a species, and it's just a very interesting way to look at this in a different sort of developmental pathway. Could yield important insights. So I wanted to thank all of my teams for the different species, and it's a whole community of people who came together to do a lot of this work, and it's been fun and interesting for all of us. And thank you all for your attention and interest.
Thank you very much. That was very interesting. We have time for a few questions, so feel free to get to the microphone.
I have one kind of random one. I know it was a very small part of your work, but as someone who works with baleen whales, I can't really go and just take a larynx home with me. So I was wondering if you could kind of describe very briefly the process where you took the larynx and then blew air through it. Like, do you have to prep it or just something about it?
Yeah, so for the ones that we did with that, the important thing is to try to deep freeze it quickly. So we would use liquid nitrogen to, so I would go into the field, collect the larynx, put it on ice, and then rush it back to the lab and deep freeze them within liquid nitrogen so that you don't have ice crystals forming within the vocal folds in the tissue, which could split them apart and disrupt things when you try to do these really, when you thaw them later and try to do these very specific manipulations. So for the excised larynx, I was just doing very preliminary things. There's whole groups who specialize strongly. Tecumseh Fitch, of course, and Christian Herbst. Yeah, I can play that playback. So yeah, there's just a whole setup that you have within your lab where you have a tube that is forcing air through, and yet it's a certain measured output, so you know how much pressure is going through that tube. And then you have to fit the larynx on top of the tube, and you just basically get a clamp or very strong zip ties, and put the trachea over the pipe that's coming up with the air, and hold it on there tight. And then you have these stabilizing structures on either side, which screw into the larynx, into the thyroid cartilage, the shield that's holding it there. And then you have to get some of the anatomy out of the way. So the epiglottis here, we had to pierce and then kind of hold up out of the way so it wasn't flopping over the larynx and interfering with things. We also had then pressure on, let's see. Things back, but then, these are also elements which are pushing the vocal folds together, and you can do that at different strengths so that you're pressing the vocal folds together so that when you force air through, it actually does the isolations. Otherwise, they're just going to hang loose, and you're not going to be able to simulate what it would be if the animal was physically closing them with their arytenoids. So I'll show what the video looks like, and maybe that'll help. And then we did, high-speed video. vary the pressure of the air going in and out, so you can see like frequency sweeps if it was an animal who was producing sort of an increase in their aerodynamic power. And then you can measure how that affects the resulting frequency. Those are the vocal folds, going like that. And then they get into this rhythm. So in Cicadere, you wouldn't see all this floppiness. It's kind of like a motorboat. Yeah. So you can measure how the air pressure and then the pressure keeping the vocal folds together affects the acoustic output. Thank you. Yes.
Anyone else?
Yeah, thank you for the talk. So about bison, when you show the different arrows about the signal process, I wonder, like in frogs, I think like the frequency indicates the mass, and mass probably is also the signals between males to see which one would win a fight. But in your graph, you showed that mass will indicate frequency, will indicate dominance. Did you, I mean, how you know this? It's going that way? Did you make some tests, or in the literature, we probably already know?
Do you mean the fundamental frequency? Or the
yes, the previous graph, if you can. No, yeah. So the Foreman frequency. So. Could it be that the formal frequency indicates the mass and not the other way around, and the mass is actually the signal of dominance, like it's in frog, for instance? If I'm not sure about frog, but probably. So how do you test it, or how you know it goes that way?
Yeah, you could think of it from the other perspective as well. We were trying to disentangle this by looking at it from the anatomy perspective, and that the anatomy is having a constraint on what the formant frequencies would be. And so, yeah, I mean, you could sort of skip this middle part of formant frequencies and say, does mass predict dominance? Is that what you're saying, like directly without looking at the acoustic parameters? Or that's not what you meant, is that?
Yeah, also, but it's probably, so bison could access to mass visually.
Exactly.
But also for the one who are far, or it could be another signal, the format frequency, another signal to access to the mass, which will be, it's a hypothesis, probably the signals that they help them to indicate dominance instead of the formant frequencies.
Well, because the formants are giving such a good indication of mass, and they still are doing a lot of rut activities during the night, and so they're not seeing this other individual. And I think you're absolutely right, because they also do the walk past each other, and they size each other up, and there's so many different dominant signals that they're not just getting this information from the calls, for sure, or just getting it from the formants. I think it's a lot of different aspects that it's building towards. But yeah, I think especially for the night, they need to get this information from somewhere. You know, they're not taking any rests at any periods, and yeah, it's interesting to disentangle what's more important when, and yeah, what you might be able to fake and not. But formants are something, yeah, it's hard to fake.
Thank you. We do not have any more time for questions, but feel free to continue the conversation with the coffee break. We're just a bit late, so please, if you can be there at 50, it would be perfect. Thank you again, Megan.
Thank you.
But let's go for the... This is Léo! And Margot will introduce the session. Thank you.
Okay, guys, let's start the last session on sound production and perception. So, Léo, welcome. We will talk about the first investigation on the behavioral context of underwater sound production in juvenile green turtles.
Okay. Hi, everybody. So I will present you the result of my first paper. I will submit maybe Monday, I'm not sure. We will see. So this work is a part of my thesis, supervised by Isabelle Charrier and Damien Chevalier. So first, why be interested in the sound production of sea turtle? We know that understanding the sensory ecology of animals, that is how they sense and respond to their environment, can be an effective tool for the conservation of endangered species, such as sea turtle. Recent studies show there is acoustic sound production in terrestrial and freshwater turtle, and some results suggest it could be acoustic communication. However, we have just a little information on the sound production of sea turtles, and this lack of knowledge is due to two factors. The first one is no one has ever thought to listening to them. And the second one is this is very difficult to collect the data in the natural environment because it's in the sea. And so, as no studies had been carried out on the subject, we published an article describing the sound repertoire produced by juvenile green turtle. However, just because they produce sound, that means that is necessary acoustic communication. And it was by researching the subject we hope to find out. We also would like to understand the context and role of different sounds, and so effectively improve the sea turtle conservation at a lower cost and effort. Thanks to technological advances, it is now possible to study sea turtle in their natural environment using biologger directly attached on the carapace or the shell of the turtle. And in this biologger, there is camera and multi-sensor integrated. So this is an example of what we can see on the video. And listen, there is some sound produced. So all the individual equipped with the biologger was in Martinique. They are all juvenile. And this is the old model of camera cats we put on the shell with the suction cup system. And this kind of sampling is similar to what is described as a focal animal survey. That is an intent tracking of an individual in order to collect all his states and events over the time. So I use Avisoft NR for my analysis. And one very useful tool for me on Avisoft is the labeling option, because I largely use label on my analysis. So here, for example, the turtle was just released from the boat. Then she produced 2 sounds. So this label, as I said, are really useful. And I use a lot of them. So 11 label of sound, 6 label of behavior, and 3 external event label in order to understand the context of sound production. So all these labels are defined by a start and a stop. So I have the duration for each sound and each event of my record. So I have the duration. and then I translate this duration thanks to the Miconi method, consisting in cutting a day in 1 second sequences. And then I will just code 0 if there is no label and 1 if there is a label. After that, I will just look for the 1 second sequences co-occurrent between sound and behavior or sound and external event. So here, for example, I have 6 1 second sequences of a sound, and 2 of them co-occurred with the behavior. So I can say 33% of the sound was co-occurrent with the behavior. So I will present you the context of the production. So my sound are classified in 5 categories. The first one is the pulse with the mono, the doublet, the triplet, and the multipulse. So the multipulse, I will play it for you. It is the same sound than the mono, doublet, and triplet. It just repeated multiple times. Okay, better. Yeah, thank you. And there is also the talk, but the talk is too low to be heard with this headphone, so I will not play it today. So all this sound mostly co-occurred with resting and swimming. There is nothing more for this one. The second category is the low amplitude call with the croak and the rumble. Here again, they mostly co-occurred with resting and swimming, but the interesting thing here is the inordinate number of rumble. As you can see, it's more than 15,000. And the most interesting part is all a large part of this song was recorded on only 4 individuals after the sunset. So we have some evidence it could be a chorus. I will tell you that later. And so, as you can see, this is a very high vocal activity. My third category is the frequency modulated sound. So this is the most original sound we have. This is the same sound you've heard during the first video. So this sound mainly co-occurred not with resting and swimming, but with scratching behavior. That is very difficult to interpret, but it's the strongest result we have for now. The fourth category is the squeaks with a short squeak. And the long squeak. So here again, this sound mainly co-occurred with resting and swimming, but the interesting fact is the long squeak was recorded on 3 individuals during a flight behavior from human. So here I have an example. So the turtle will surface to breathe. Then she will find herself facing the beach. And then she will die very quickly. Like that. And now you have a little long squeak. And if we go back, I'm not sure to... I cannot choose... Oh yeah, okay. So when the turtle dive, in fact, the three suction cup on the 4 detach from the shell, and the camera completely go back. Now we can count 1, 2, 3 swimmers. Probably the turtle just must feel trapped between the diver and the land, and so she starts to flight. And during the flight, she produced the long squeak. This one. Okay. And so the last category is the grunt. This sound is once again mostly occurring with resting and swimming behavior. But this is the only sound which is associated with a stereotyped physical movement. This is a retreat head movement. So here the turtle is in vigilance, and she produces a grunt. It's very visual, and in this one, the turtle approaching a core specific, and she emitted it. So to conclude, we recorded for now 4 sounds in a specific context: the FMS during scratching, the long squeak during flight behavior, the grunt, which coinciding with the retreat head movement. We think it could be a potential visual agonistic component analogous to aggressive threat signals. And the chorus of rumble during resting after sunset. For this chorus, we have several clues indicate that rumble were not produced by a single individual, but by several individuals close to the biologger. And so it could be maybe the first evidence of acoustic communication, but I have to work a bit more to understand it. And surprisingly, we had no specific sound during physical interaction between conspecific. Maybe just because they use other medium to transmitting information, or maybe they just don't need to transmit information in this case. So after that, we wish to understand the biological function of this sound. So there is a lot of work for this. I will continue the analysis on the recently equipped hawksbill turtles. This is another species. And also equip adult green turtle to compare our result, because these ones are only juvenile green turtles. So thank you for your attention.
Any questions, guys?
Thank you for your very interesting presentation. I have 2 questions. First one, do you think there may be some production during feeding attempts by turtles? I don't know how turtles are foraging. And second, what do you think may be the aim of this night chorus for turtles? Thank you.
Okay, so for the first question, no, there is no particular sound during foraging. We could hear the scrunch on the algae, but no sound except by product. And for the chorus, this is the subject of my second paper. But for now, and for several things that I found in literature, the chorus for me could be an acoustic signal to warn a predator, that is mostly sharks, during night. They are vigilant, and so as the sharks does not hunt any prey in vigilance, if the turtle warn we are vigilant, maybe the shark will not attack the turtle.
Do you have evidences or reason to think that they are producing sounds that may relate? Oh yeah, you mentioned the flight sounds. So they could produce this sound in presence of predators.
Maybe, but we have no predator during the day, and so we not record it. So for now, it's a bit difficult to answer.
Anyone else? No one.
Thank you.
Thank you, Léo. Okay, the next presentation, oh là, pardon, je suis pas au bon endroit, is Naïs, and she presents sound and vibration control crocodile hatching.
Thank you, Mathilde. And I will just start to say a big thank to other organizing team for this Congress, and also to you that stayed this Friday afternoon to listen to the last presentation. So I'm in the last year of my thesis on the perception of sound and vibration in crocodiles generally, but today I will present how sound and vibration control crocodile hatching. So the birth of a baby or the hatching of an egg are moments when the risk of predation is particularly high, especially in oviparous species where clutches contain many eggs. And one strategy for limiting this predation is based on mechanisms that synchronize the hatching, which reduces the risk, the individual risk of being targeted, and which allow hatchling to emerge under parental protection. And so, despite the biological significance of this process, all the mechanisms involved in hatching synchronization are poorly understood. So there is previous researches that work on the hatching synchronization and the impact of sound and vibration, how the stimuli could have an effect on this synchronization. And there is a study on birds, insects, but also turtles. And for example, for the pig-nosed turtles, we saw that all the embryo tested with substrate vibration hatched together, and suggesting that the hatchling could react and could detect and react the signals of their nest mates in the nest and hatch accordingly. And in the present study, we investigate the mechanism governing hatching synchronization in crocodilian. So crocodiles are animals characterized by a high predation pressure on youngs, but also an extensive parental care. So, for example, the female are really involved in the protection of their young, and they invest energy in their brood, and they lay several dozen of eggs in a nest made of a pillowed vegetation or dug in the sand. Then she will guard the nest during the whole incubation period, and at the end of this period, she will dig out the nest to liberate the young and to help them to hatch by grasping them in her mouth and carrying them to the nearest water. And this digging behavior is induced by hatching calls produced by the young mature embryo. And these hatching calls also elicit response from the embryo, and they react by vocalizing themselves in response of this call. So this hatching call appears to act both as inducer of parental response, but also to synchronize the hatching of the young. And some authors suggested that there are also vibrational signals that could be involved in this mechanism, but there is no study for the moment in crocodilians. So here it takes the effect of airborne sounds and vibration on the hatching in crocodiles, in mature Nile crocodiles in the Croco Park of Agadir. So we tested 30 embryos, but 6 were found to be non-viable at the end of experiment, and so were removed from the sample. As the sound stimuli, we used hatching call, so in sequences of 1 minute of hatching call with between 2 and 3 calls per second. And this call has been recorded prior the experiment from mature embryo ready to hatch. And for something like this. And for the vibratory stimuli, we reproduce scratches and impact that reproduce the movement inside the embryo when the embryo starts to hatch, but also the movement of the mother that is digging out the nest. So this is also sequences of 1 minute. So this is the setup. We have here the loudspeaker and the vibratory devices with the egg fixed on a support. And we have 5 different conditions. We have the vibration condition only, the hatching call condition. We have the vibration plus hatching call condition to see if we could have synergic effect between the 2 stimuli. But has the vibratory device also emit a sound when transmitting the vibration? We had also a control condition where we play back only the sound produced by the vibratory device. And we have also a last condition, silence condition, where the embryo were just transported to the experiment room exactly in the same way as the other, but we play back nothing. So this is the experimental timeline. We carefully transport each egg once a day. Each egg is stimulated with always the same stimulation once a day, and is carefully transported to the experiment room, then fixed to the support. We systematically waited 5 minutes before sending the stimulation. Then stimulation was applied during 1 minute, followed by a 10-minute observation period. And then the egg was returned back to the incubator. So for each trial, the egg behavioral response was assessed by assessing 1 or 0, whether we observed each of these 3 behaviors, so movement, vocalization, and hatching. And so we calculated first the probability of each of these behavior. So we see for the probability of movement that the tested embryo responded to each of the 3 experimental conditions, so vibration plus hatching call and hatching call, by with motor response, but this is a vibration condition that elicits the strongest motor response. For the probability of vocalization, embryo responded by vocalizing also to the 3 experimental conditions, but it is the hatching call condition that elicits also the more important vocal rate from the embryo. And for the probability of hatching, it's not as clear. And we see that embryo stimulated with vibration and hatching call respond by hatching to the stimulation, but we do not clearly understand why we have this situation for vibration plus hatching call, and we have no clear difference, for example, between this condition and the control condition. So maybe it is not a real natural stimuli for them, but they respond clearly to vibrational signals. So this is a little video, an example of egg stimulated with vibration. We see that is starting to crack the eggshell with his snout. And it could take really a long time for a baby to hatch. And this was just after the vibration stimulation. We also calculate the cumulative percentage of eggs for each behavior. So in this graph, the darker is the color, the percentage, the more high is the percentage, sorry. And we see that in this figure, we just see that embryo tends to hatch earlier when stimulated with the vibrational stimuli compared to the other one, and that exposure to hatching call don't have this comparable effect. So by conducting playback experiment with this mature Nile crocodile embryo, we see that in addition to hatching call, there is also the vibration stimuli to take into account when study this whole mechanism of hatching synchronization, and that the vibration tend to synchronize also the hatching, but that embryo also hatch earlier when exposed to vibration compared to the other condition. And so the crocodile nest could be seen like a real communication network with sound and vibration stimuli that lead the protagonist to decide on the right moment to hatch, with the parent taking really an active part in this process. So thank you for listening, and if you have questions, I can answer.
Okay, we have the time for questions, so if you have.
Do the mother also interact, also call and have an interaction with the embryos? Because, for example, in birds, they do like hot, I forgot how they call it, but to warn that the eggs that it's too hot outside, so it will like they will hatch quicker. Is it similar in crocodile, or is it just in between embryos?
No, the mother also reacts to the embryo vocalization, but they don't vocalize, and they don't vocalize with his embryo. She's just guarding the nest, and for example, when it is too hot, she will really stay on the nest to protect them from the sun. But all the mothers are really involved in this process of guarding the nest. And in another study, we studied the impact of the amount of hatching calls produced by the embryo on the behavior response of the females. And we saw that the females really wait for a continuous series of hatching calls to begin his digging behavior. But there is no vocal interaction from the mother.
Other question? Yes?
Hi, thank you for the talk. I was just wondering if you took some kind of some genetic sexing of the hatchlings, because I've read somewhere that the eggs, like the female embryos, hatch in warmer temperatures or something like this, faster. So, do you know something about that?
Yes, we didn't made any genetic analysis, but in our experiment, all the embryos were in the same incubator at the same temperature. So there is really no difference between our eggs, and all eggs are from the same clutch of the same female. So
okay.
We have no more indication about the genetic.
Okay, thank you.
I have one. Do you know if they communicate after with vibration, the mother and the babies, after hatching?
After hatching?
Yeah.
There is some example of vibrational communication, but in adults during the courtship, where the males produce water dance. When they produce subaudible vibrations that lead to water dance just above the back in the water, and it's all water droplets just above the back to attract females. But really vibrational communication, I don't think so. But they used, that's sure that they use really vibration information to have more information about their environment, as they have a lot of sensory mechanoreceptors that are really, really sensitive. But really communication, I just have the example of this subaudible vibration in males.
Other ones? No? Another one.
Just wonder, how did you make the stimulus? Like you say that you managed to reproduce the vibrations of the egg and the mother digging. So how did you do that?
We take a hen egg and we reproduce the movement with a clamp of scratching and impact, and we record it with recorder. And when we play back this to the vibration devices, it reproduces the same vibration when the egg is really mechanically linked with the support. Thank you.
Thank you. Thank you, Naïs. Okay, so next presentation is Stefano Gonan. So he will talk about the bond for sounds, innate cognitive and emotional predisposition in domestic chicks.
Good afternoon, everyone. My name is Stefano Gonan, and I am a PhD student at the Laboratory of Animal Cognition of the University of Trieste in Italy. And today, I'm going to take you on a journey into the world of auditory perception. And our story begins inside a small egg, where a domestic chick is preparing to hatch. And this chick is surrounded by a symphony of different sounds that are continuously stimulating the young chick's auditory system. But which of these sounds, which of these acoustic stimuli will capture the chick's attention? So are there some innate preferences for certain acoustic features or not? To investigate this, we decided to perform an experiment with 3-day-old domestic chicks using as the turning bias paradigm. That is, we mainly focused on the head movements of the animal in response to different acoustic stimuli. And so we tested 121 domestic chicks in 2 different experiments inside a soundproof arena, such as that depicted in the image. And the sounds were played from 2 loudspeakers that were placed at the ends of this rectangular arena, while each chick was placed inside this small cage with a frontal opening for feeding. And we registered the behavior of the animal through a camcorder and then analyzed the head movements and saw the behavioral responses of all the animals through professional behavior analysis software such as Boris. And so our hypothesis here was very simple. That is, chicks would stop eating and listening longer to those sounds that they found more salient and interesting compared to less engaging stimuli. So, trying to hear the world from a chick's perspective, we ask ourselves, what makes a sound interesting to a chick? So are there acoustic predisposition to pay more attention to certain calls, to certain vocalization and acoustic features than others? So to explore this question, we used as auditory stimuli the domestic chick's entire vocal repertoire, including both positively and negatively emotionally valenced calls, as well as the adult call vocalization. And so let's have a listen. The first one is the distress call, which is emitted by domestic chicks when they are isolated from other siblings or from the mother. So it's a negative valence call. Then we have a positively valenced emotional call, that is the brood call, that is mainly emitted when domestic chicks are near other siblings as a mean of keeping contact with them. Then we have another negatively valenced vocalization, that is the fear trill, which is kind of particular because it's emitted both when chicks are frightened, but also when they are negatively surprised by something or someone. And it has this strong frequency modulation if compared to all of the others. And then we have the food calls, and as the name implies, is emitted when there's a presence of food resources. In the end, we have the adult cluck, that is the typical vocalizations emitted by chickens when they are exploring their surroundings. Okay. So what did we discover in the first experiment with natural calls? Well, as you can see from this point range graph, one sound in particular stood out, that is the fear trill. So that is, the animals tested spent significantly more time listening to this type of vocalization than to others. And this suggests that maybe the species possess a sort of innate ability, an innate mechanism to allocate their attentional resources to those sounds that are signaling the presence of a potential threat. And the interesting thing here is also that they seem to ignore the distress call and also the adult cluck. In the first case, maybe because of the fact that the distress call is emitted as a vocalization to signal the discomfort of separation from the mother. So it's a type of vocalization that is mainly directed to the hen, not to other siblings. That's the reason why they seems to ignore this type of vocalization. Well, for the adult cluck is quite puzzling, because we thought that, well, the adult should be some an attention grabber vocalization, but maybe the bond between the chicks and the sound of the voice of their mother is something that is more learned than an acoustic predisposition from the very first day of life. And we then ask ourselves, can we trick the chicks' ears? In other words, how does the perception of these vocalizations can change when the timbre is varied? So is there always an innate tendency to pay more attention, to allocate your attentional resources to certain sounds and certain acoustic features, or the variation in timbre can also modify this behavioral response? So to trick the chicks' ears, we used tone transfer, that is a deep learning algorithm developed by Google Labs, and is peculiar because this algorithm is able to change the timbre of sounds, and we use that to transform both the distress call and the fear trill into the timbre of two different musical instruments, which are the trumpet and the flute. So here we have the distress trumpet. Well, here's the distress flute. And the same for the fear trumpet. And the fear flute. So all of the acoustic features that are somewhat related to timbre are changed, but the temporal structure of these sounds is unchanged in regard to the original natural calls. And what did we find out in this second experiment? Well, as can be seen again from another point range graph, it seems that the trumpets bold sound mimic the urgency of the original fear trill better than the softer tones of the flute. But the very interesting thing here is that this effect seems to be vocalization dependent, because we have this strong difference in the time that the chicks spend listening to the different sounds only for what concerns the fear trill, but not the transformed distress signal. So, what acoustic features could explain the observed behavior of domestic chicks? Well, to investigate this, we performed a partially square regression on 22 acoustic variables that were previously extracted using the analyze function of the SoundGen packages of RStudio. And we found out that the response of domestic chicks to natural calls is essentially predicted by high duration, entropy, and spectrum variation, as you can see from this graph, where you have the point ranges that, if they are quite distant from the center red line, are considered significant. And by a low change in acoustic features such as pitch, amplitude, and harmonicity. But on the contrary, when exposed to sounds produced by musical instruments, there seems to be a shift in those acoustic features that can predict these behavioral results. That is, the chick's attention seems to be captured by sounds with a high harmonic-to-noise ratio, harmonic energy, and also by a higher pitch, but also with a low change in all of these significant acoustic features. So, what can we conclude from our experiments? Well, the first take-home message is that our results clearly demonstrate that domestic chicks process and perceive sounds with distinguishable timbres differently. In particular, for what concerns species-specific timbres, chicks appear to have an innate tendency, an innate mechanism to prefer and also to attain those sounds that have a greater unpredictability and complexity, that are all two features that also are able to reduce the risk of habituation in the listener. But at the same time, those unpredictable and complex sounds must have a clear tonal structure, so clear tonal elements, maybe because these features can make the sound more recognizable or at least easier to process for the listener. While in contrast, for musical timbres, we have a different situation. And for those sounds which are also entirely unfamiliar to domestic chicks, there is a clear tendency to pay more attention to features related to both harmonicity and pitch. And mainly because these features give the sound a more voice-like structure. So it seems that maybe for what concerns those sounds that are completely unfamiliar to animals, domestic chicks are trying to search for those sounds that in some sense are able to mimic the sound of a voice, and so are more attentive to these features. So, before concluding, I would like to extend my gratitude to our team, especially to Andrea Di Segna, to Professor Giorgio Valortigara and Professor Cinzia Chiandetti, and obviously to all the CHICS participants who made this study possible. And I would also like to thank you for your kind attention.
Thank you. Some question? We have the time. So, thank you.
Thank you.
And for the last talk of these conferences, the last presentation with Blanche, and she presents Do Ship Converts.
So good afternoon, everyone. I'm Blanche, and I'm going to talk about the conversations among sheep to conclude this marvelous event. So first of all, you have to know that human language is characterized by the production of vocal interactions that obey temporal and social rules, which which will allow the expression of cooperative behaviors within social groups. And for instance, is in this public, we all come from different countries and we all may speak different language, but we share some similar rules that are universal in our language. For instance, the vocal turn-taking. But what is it? This is a reciprocal exchange of alternating short and flexible turns between interactants based on specific properties and some coordination principles. It will avoid the overlap of the vocal signals between the interactants and also minimize the response delay between the interactants in order to maximize the quality of the information that are transmitted. But the question in the vocal thinking is to know if is it uniquely human or if we can find it in other animals. And recent studies shown that they found vocal tone-taking in some birds, insects, anerones, and some species of mammals. And in my study, we were wondering, what about the species that is less known to have some sophisticated cooperative behaviors? That's why we chose the sheep, because it hasn't been studied yet, and it's a mammal that lives in groups and is less known to have sophisticated cooperative behaviors. So my main questions were, what are the different types of vocal production that we can find in sheep? And if we have some vocal tone-taking, which is a type of vocal production, is it correlated to the spatial proximity between the interactants? The spatial proximity here is used as a proxy of the social affinity. So my hypothesis were due to the fact that sheep is less known to have some sophisticated cooperative behaviors, the existence of vocal thinking is unlikely. But if there is, it could be linked to the spatial proximity or the kinship between the interactants. So how did we do this? I was in the INRAE Center Val de Loire, and I had 28 ewes that I distributed into 4 groups, and in each group there are 2 sisters. For the experiments, 1 sister were taken in the corridor with one of her congeners for 10 minutes. So it looked like this. Oh, this, and it sounds like this. So sorry. So I did 20 isolation sessions per group, so in total 80 sessions. And after that, I was on Pratt software to measure the duration of each cry. So until now, I only had the time to analyze 4 sessions per group, so only 16. And so this is, for instance, a call and its duration. And And it's useful because we can determine, after all, the intercall duration, which is the duration between 2 consecutive calls. So it will be this. And thanks to that, we could determine some patterns of vocal production. And for instance, there is the chorus, which is more than 2 individuals are vocalizing at the same time. It sounds like this. There is also the vocal tone-taking, so it is a vocal exchanges without overlap. There is also overlapping calls. It's vocal exchanges with overlap. So the overlap is when the intercall duration is negative. For instance, just here, this call begins while the previous one isn't finished. So there is overlap of the calls. And after there is isolated calls. It's when one individual will vocalize and it will have no answer from other members. And there is also repeated calls, so it's one individual that will vocalize several times in a row, like this. So So after, to know if the vocal tone taking was linked to some spatial proximity, I used scan sampling in order to note 1 time a day for 1 hour each 5 minutes the spatial position of each individual in its box, in order to know what is the identity of its closest neighbor. Because if there are some preference with individuals and that could show some social affinity, could we link it to some vocal tone taking where they could also have some preferences for the interlocutors between the use. So what did we found? We found some vocal tone taking among use. So as you can see here, the intercall duration is negative, so we could determine 28% of overlapping calls. And after, we had to know which maximum intercall duration we could use to define what is vocal tone taking. So we used the method that is used in previous studies, and it's based on the last maximum that will be above the dotted line, which represents the mean of occurrences. So it's an arbitrary value, but with that, we could consider that if the intercall duration is superior to 4.2 seconds, then we could no longer consider it that we have vocal tone taking. But with this, we have 54% of vocal tone taking in our data set. And after, if the intercall duration is superior, so we have, for instance, isolated calls, which are non-exchange calls. And on this histogram, just to show you all the proportion of the vocal production patterns, and we can clearly see that vocal tone taking is the majority of our calls. What about the spatial proximity? So here on this matrix, it's the results for the blue group. You have to know that it's representing of the results of the other groups. And we can see clearly that when the intensity of the color is darker, it means that the spatial proximity is higher, and so that maybe there is some strong social links between the individuals. And so, for instance, the individuals named 4B was a lot in contact with the individual named 2B, whereas 5B and 1B weren't close at all. And after, for the vocal affinity, so this is preliminary data because I only had the time to analyze 4 sessions. So we can first see that there are some vocal interactions that are missing, symbolized by the gray cross. But we also can see that there are some vocal interactions between the 2 sisters, in yellow, so the 5B and the 6B. And the other dominant vocal interactions are between the U's that were isolated together during the experiments. So for instance, the 5B and the 7B, the 6B, 7B, and same with the 2B. When I will finish all the analysis, it will be interesting to know if there is a correlation between the spatial proximity and the vocal affinity. So I didn't have the time, but I just wanted to see what I had, but it has no significant meaning. But for instance, just to know, there isn't, with the core test, there isn't any correlation between our 2 conditions, but it was expected. So to conclude, our hypotheses are rejected because we did find the presence of vocal tone taking among sheep, which we are happily surprised because it wasn't demonstrated until now. It's interesting also because, for instance, in bonobos, the ratios of vocal tone taking and overlapping calls are similar to the one that we find in sheep. But for instance, in chimpanzees, it's only 6% of vocal tone taking. After we saw that we had some spatial proximity that could be explained with some social affinity or some rank dominance, and to finish, it was expected, but we had no correlation. So these are promising results, and it's interesting to continue the analysis. The major limit of this study is that we use stressful stressful condition. And when I talked earlier about the bonobos and the chimpanzees, the studies were based on spontaneous vocal production from the animals. So the stressful condition here is justified due to the fact that sheep doesn't vocalize much, whereas they are in a context of isolation, danger, mother-lamb relationship, or if the food is delayed. And so it's really interesting to do these experiments, but with the spontaneous calls of the sheep. And if these results are confirmed, sheep may use similar rules that guide human conversation, so it's pretty cool. And this study may provide valuable information of the role of group life in vocal production and importance of cooperative behaviors, because this study could also show and suggest that vocal turn-taking could be used in another context of cooperation, and it has to be determined. So I would like to thank Florence Levrero and Florian Fournier for their help on this study. I would also want to thank Mathieu Keller and all the UEPAO team from the center in RAE, and also thanks the committee of the GGBA for their super work, and also you to be there and for your attention. Thank you.
Okay, some question? Yes?
Hi, thank you for the talk. You said that you found some turn-taking, but could you explain a little bit more how you decided that it was actually turn-taking? Did you focus on one individual at a time and then looked at how much it was alternating with another one? Or just that the fact that the calls were in alternation and that this was more than random, it was actually the group alternating and thus turn-taking?
For the moment, it's just between the alternance of the calls, like on the histogram. So because they are not overlapping and they were consecutive with the duration, the intercall duration. Does that answer to your question?
Yeah, no, I was just, because I also worked on turn-taking, and I found that it was important to say that it was more than random, to say that they were actually doing turn-taking, but I guess that's what you're doing. So.
Yeah, and I think we will never know what they are really saying in a biological sense, but we can just determine that it wasn't overlapping, it wasn't a chorus, so it could be that they are listening to each other with big... but so...
Thank you so much.
Thank you.
One more question, maybe?
Just wondered, what is the age structure of this group? Are they all the same age, or are there... you mentioned sisters. Are there other relationships, I guess?
Okay, so in each group has the same composition. There are 2 sisters, 3 individuals that are not linked at all between them and between the others, and there are also the or maybe 4. Wait, I will just take my. Yeah. So there are the 2 sisters. There are 4 individuals that are not linked, and there is, there are the 2 sisters. All the others are not linked, and there is also one older you, which is like 4 years old more than the others, and the others are like 1 year old.
Okay.
And so between the yous that are sisters, it's the same age also, the same. Born the same moment.
Okay. Thank you. Yeah, I was just interested if there was kind of grouping going on? Like you mentioned the mother-lamb communication. Yes, that was what I was interested in.
Yeah, no, there isn't lamb in this experiments.
Thank you.
Thank you.
I have 2 questions. So you said that the proximity is like a good proxy for the relationship. Do you know if there is like other behaviors, like for example, scratching each other or stuff like that? Or?
Yeah. I didn't quantify them, but just on spontaneous observations, they have some aggressive behaviors where, for instance, when they eat, if one, and she may be the dominant, but I don't know, and she wants the place of another one, she will go back and she will push it and jump on it. And so if the one who is doing that is the dominant or has more place in the group, the other one will move. So there is that. They can also do like a fight with the head. They can also eat some grass that is on them, but on the other. Sometimes the other don't really like it. And they also sleep sometimes close between each other. And yeah.
So do you think you will take it into account later, like to?
I didn't quantify it because
you don't have videos.
Yeah, but it could be interesting to link, because also in vocal turn taking, what is interesting is that there is the response delay, so the intercall duration, but there is also the direction of the gaze and the direction of the body to know where you will take the turn or not. And so this could also be interesting to describe this kind of behaviors.
And also related to the question about to see if it's above random, did you try to do a permutation thing where you take like the, in Pratt you have like for each individual you have like when they start the call onset, and you take that series and you randomize it with different series from different moments, and you see if there is less overlap than with the actual interaction. With the actual, when you recorded it. It's a technique that has been used with seals. It's a pretty cool technique. So I don't know if you knew about it or tried.
I didn't do it yet, but I just used in one session just all the calls of one session to know that.
Okay.
But it could be interesting.
Yeah,
Thank you.
We can talk about it.
It's almost finished.
Sorry. Do you plan on looking at the call types? Maybe there aren't any differences, but I guess because you're putting them in a distress kind of situation, they could be vocalizing differently if between the ones that are inside and the ones that are outside, compared to the ones that are both outside. And also, just maybe looking at the length of the call as well.
Yeah, I would love to do that when I will finish all the analysis of the 80 sessions. But it's really interesting because just on my idea on spontaneous observation, when they were isolated in the corridor, they were more doing isolated calls and repeated calls that could show maybe the emotional state of the individuals. Yeah. So it could be really interesting, yeah.
And maybe, but I don't know if that's possible, maybe you could try also to see what happens when you reunite them.
Yeah, it's quite difficult, so I won't use my data for that, but sometimes it was really interesting because once I opened the door of the box and they reunite, no more sounds. So it was cool.
Cool, thank you so much.
Thank you.
Okay, thank you, Blanche.
Thank you.
Okay, clap de fin. Thank you all so much for coming to Brest with this beautiful weather, this beautiful sea. Really glad we could all meet. We could all exchange on your topics, getting to meet each other. Quick note, if you guys want to take some of the leftovers, feel free to do so, because otherwise it's maybe ending up in the trash. And if some of you are keen on helping packing up this place before you leave, don't hesitate to stay a little bit and, you know, pile the chairs and move the poster grille. Voilà. Thank you very, very much.